Louise. I am the Senior Innovation Officer of the Innovation Center of Denmark in Tel Aviv, specialized in life science, healthcare, and AI. And I want to thank you all for joining us this morning to our webinar on bioconvergence and multidisciplinary innovation in life science. In collaboration with our colleagues at the Danish Consulate in Istanbul, as well as the Medicon Valley Alliance and Israel Innovation Authority. This is a rapidly emerging field with interesting potentials, which we want to explore this morning. So I want to thank all the speakers from Denmark and Israel for joining us. All of those participating will have an opportunity to ask questions to the speakers during the webinar. So please just post them in the chat, and then we will post them to the participant during the panel discussion at the end. And first, I would like to pass on the word to Anne Dorte Rigelsen, our ambassador of Denmark to Israel. So please, Anne Dorte.
Thank you very much, Louise. Thank you for the invitation to participate here in this very, very interesting panel. A special thanks, of course, to our partners in Medicon Valley Alliance and Israel Innovation Authority. And as Louise said, to the speakers who have joined us today and to our partners in Turkey, in Istanbul, it is lovely to see that not only Israel in the Abraham Accords, but also Denmark in this triangular can do what is necessary for the research, namely cooperate. As many other fields, life science and healthcare have undergone rapid development and transformation in recent years with the application of new approaches and tools to medicine, such as artificial intelligence, machine learning, and big data. A development accelerated even further by COVID-19, emphasizing the need for efficient and effective drug development and development of healthcare technologies to meet current and future healthcare challenges. Scientists are now taking the next step in building approaches to converging the fields of biology and engineering to enable new discovery in 3D printing, genetics, material, nanorobotics, and other developments, allowing us to take precision medicine and personalized medicine to new heights. Preliminary developments in bioconvergence and are encouraging and stress more than ever the importance and essence of cooperation and co-development, both naturally and internationally. Like Israel, Denmark has emerged as a global life science and healthcare powerhouse. And usually Danes are modest, but not as regards life science. Denmark is home to multinational strongholds such as Novo Nordisk, Lundbeck, GenMat, and also to an increasing number of groundbreaking Danish startups. Danish companies are internationally recognized as pioneers in disruptive research and development of new drugs, technologies, and services advancing prevention, diagnostics, and treatments of patients around the globe. Naturally backed by cutting-edge research from Danish Technical University and other excelling Danish universities and research hospitals. At the core of the success of the Danish life science industry is the strong collaboration between all parts of the Danish life science ecosystem, as signified by the creation of the Medicon Valley and the Medicon Valley Alliance. This makes Denmark a highly attractive partner in multidisciplinary life science innovation. The Innovation Center of Denmark in Tel Aviv is here to help Danish researchers and companies gain access to international partnerships for knowledge transfer, research cooperation, and technology development in multidisciplinary life science innovation. So are the Danish missions in Turkey and in the rest of our southeastern European region. We have seen fruitful research and development collaborations in life science between Denmark and Israel, and we see great potentials in doing this in the multidisciplinary collaborations between all the actors that I just mentioned. We are excited to explore the how, the what, and the why today with you. And with these words as introduction, I would like to give the floor to the next speaker, which is Søren Reinholdt, Chairman of the Medicon Valley Alliance and CEO of Macrophage Pharma. Søren, the floor is yours.
Thanks a lot. And I hope you can all see my screen. It's a great pleasure and an honor to be invited to welcome you all to this Bioconvergence seminar. It's a pleasure for me. It's a pleasure for Medicon Valley Alliance to co-host this hopefully fruitful and groundbreaking seminar. Let's just spend a couple of minutes on Medicon Valley Alliance. Why is Medicon Valley Alliance the perfect partner for bioconvergence and multidisciplinary international collaboration? First of all, Medicon Valley Alliance is by its birth already multidisciplinary. It's cross-border as the region encompasses Copenhagen, Zealand, and also the southern part of Sweden. So we say we have one cluster, 2 countries, 3 administrative regions with 4 million inhabitants. So 1, 2, 3, 4, that's Medicon Valley. So why is it interesting to do work in Medicon Valley Alliance or Medicon Valley? We see the region as a Scandinavian Kendall Square, sort of the hotbed for innovation in Cambridge, Massachusetts. And why do we claim this? We claim this because we have all the ingredients for a fantastic cluster. We have world-renowned universities like Denmark Technical University, which is represented today, University of Copenhagen, University of Lund, University of Malmö. We have actually a strong pharma presence, especially on the Danish side. And we also have the pharma industry reconverging in Sweden. We have a strong biotech industry with both scale up and startup, both in Denmark and Sweden. And then importantly, we have a lot of tech companies, both in fintech, but definitely also in AI and health tech. And we really see that this is the next frontier of biomedical research in the region. I think that is probably often overlooked when you look at the life science industry in Medicon Valley, is not only the device industry such as Coloplast or Ambu, but if you actually look at companies like Novo Nordisk, you have a company that in sort of basic terms is actually doing bioconvergence by not only doing pharmaceuticals, but having a very strong focus on engineering in their devices, and also recently in integrating AI and digitalization in both their front end, but also in their back end products. So the region has all the ingredients to be a hotbed for bioconvergence. If we take a look at the workforce, we have approximately 7,000 life science workers with approximately 3,500 international students. and add to that another 2,500 international top researchers working in the region. That has led over the years for the region to become at top of the range in innovation, here represented by the European Innovation Scoreboard, where you can see that both Sweden, Denmark are in the top 4 as being very, very strong innovators. And if you actually allow yourself to zoom in on the graph here, on the picture here, you can see especially the region around the capital region Copenhagen and southern Sweden are among the strongest innovators in the region as well. So a multidisciplinary and multifactorial network with pharma, hospitals, biotech and tech, a strong and well-educated workforce has reflected and cemented itself in a strong innovative network. So when we look in Medicon Valley Alliance, what is the next step for the region, having established the academia, the research institutions, the hospitals, the biotech, and the pharma? We really see, as I said before, that not only looking to devices, but also to all the bioconvergence that we see in manufacturing, from both biologics and small molecules to health tech to digital AI, is really sort of the perfect launch ramps for bioconvergence. And in Medicon Valley Alliance, as an organization, we are trying to really focus on and strengthening of that, and we are doing that on a couple of so-called networks that everybody that is member of the organizations are free to attend. We have a microbiome network that is, of course, working on the microbiome, trying to connect researchers, academics, and industry together. We have our MedTech network that works across borders. And what we will do in 2021 is to start interconnecting these networks to really sort of support structures in the region to help foster further research in bioconvergence. A good place to start could be MedTech and microbiome network. So with this short introduction to Medicon Valley Alliance, I'll give you a view over a number of our members, and then I'll wish you a fruitful day, a good discussion, and hopefully within a couple of years we will see the first tangible outcomes of today's deliberation. So once again, thank you, and have a fantastic conference.
Thank you very much, Søren, for your insight into the Danish life science sector. And as we see and hear both from the ambassador and Søren, Denmark is a strong and reliable partner in the bioconvergence and multidisciplinary innovation field. And now I would like to turn over the word to Itai, from the Israel Innovation Authority, who will give us an insight into what's going on in bioconvergence in Israel. So please, Itai.
Itai, you are on mute.
So thank you we are very excited to be here. This bioconvergence emerging approach, emerging industry require multidisciplinary knowledge and infrastructures. And we see the Danish ecosystem is complementary to Israel ecosystem. We need to work together to develop this field in terms of human capital, in terms of capabilities in development, manufacturing, and sharing knowledge and ideas to develop this new wave of innovation. I'll take you briefly through our development of this national program. First, one word about the Innovation Authority. So this is we are an agency, a government agency. Our mandate is to fuel technology innovation into the Israeli ecosystem. We have a budget of about $500 million annually. About 1/3 of these budgets go to life sciences. And in the last 3 years, we did an in-depth analysis and rethink how to leverage Israel's strengths in science and technology in life science, how to leverage these capabilities into build health tech industry. And then we basically developed this bioconvergence approach, meaning that we see in the next several years, we see convergence of development of advances in life science, in drug development, in advanced therapies, in genetics. But this intersection into development, accelerating of development in engineering, computing, material science, robotics, and we see that this now is merging into one approach, one sector that we name it bioconvergence. The definition of bioconvergence is basically that the life science is in the center. If you look on the 20th century, where the physics and engineering were the main disciplines that transform the industry. Today, we see that the life science, the biology, is going to join and going to be the main, the pivotal discipline that intersecting with mathematics, with physics, with electronics, nanotechnology, computation, and to build a new approach, new fields that impact many industries, not just healthcare. So we see that the McKinsey report this year published that map the bioconvergence economy impact, and this impact is enormous. We see $4 trillion markets in the next every year, next to 20 years. We see about 60% of the world physical input could be made using biological means. 30% of the R&D expenses would go to life science or biological R&D. I think what is amazing is that this industry, this approach will not impact only health tech. It's going to impact many other industries like agriculture, like food tech, material, energy, even defense industry. We see it penetrating to many different industries, and that's why it's very interesting to support and to develop the infrastructures and the knowledge of these fields. We can see three main impacts of this bioconvergence trend. First of all, how to industrialize the way we do in drug discovery, the way we do in drug development, and the way we are doing clinical trials. So this is maybe the main benefits for the biopharma industry. We want to see that cell and gene therapy, the advanced therapies, are going to be the mainstream. How to engineer delivery system, how to enhance functionality and manufacturing. Design nature, how to engineer, how to program biology, talking about living machine, nanorobotics, advanced diagnostics. All these three pillars are going to be impacted by developing this bioconvergence approach. So we must say several words on the COVID-19. So this crisis demonstrates the crucial need for new innovation, technology innovation. We see advances in diagnostics. We're talking about smart protective equipment. We see we're looking for agile vaccine platforms, advanced biosensors. All of these applications and are based on intersecting of biology, science with engineering and biorobotics. So where is Israel in all this story? So we recognize this approach, this new wave. Israel have all the ingredients to be a significant player in this field. So we have an outstanding research institutes, universities, the Weizmann Institutes, the Technion, all of the many other that have top science and many factors that convergence field. We have a high-tech. We have an established high-tech industry with more than $40 billion exports annually, with AI ecosystem, advanced ecosystem, with ICT advanced ecosystem, a lot of entrepreneurs and very strong companies. We have an established medtech industry and a very advanced health system that can support the regulation and the clinical trials in this approach. But the idea is to intersect and to build an ecosystem, highly integrated ecosystem that work together to develop highly integrated products and therapies. So the vision is to build in Israeli advanced world-leading cluster that attracts talent, attract investments, and multinational companies to build and to invest in R&D in Israel. And as I said in the beginning, we're looking for collaboration with other countries like Denmark, because we understand that there is a need for complementary knowledge and infrastructure to build this very complex ecosystem. We have a program under development. I don't get into the details of this program. We have a lot of activities, but the main idea is you have two legs. One is developing science and technology infrastructures in academia, in medical centers, and how to increase the deal flow from this ecosystem to build new companies and to build manufacturing in Israel, and then to how to leverage this science and assets to build large companies and to build manufacturing, new regulation, and global collaboration with other countries. Maybe I'll take Karina to describe in more details what we did in the last year and how to plan to move to 2021. Please, Karina.
Yes. Thank you, Itai, and good morning, everybody. It's really exciting to be here after such a long effort, and thank you, Ruiz, for this opportunity, and the ambassador and all of you here. We are really looking forward for collaboration with Danish companies, Danish academia, Danish entrepreneurs on a national level. So I'm really hopeful that the outcomes of this event would be fruitful and soon enough. Just to brief you about what we've done in the last year when we started promoting this approach of bioconvergence in Israel on a national level. The Israeli Innovation Authority allocated around $30 million for this approach to promote this area over the year, and we've done a lot of activities around it. One was an open call for academic bioconvergence applied research projects that we've opened. We have an innovation lab in the establishment phase now. Hopefully it's going to be up and running in the next year. We have a consortia between academia and industry in the area of CRISPR. And we have additional, hopefully 2 new consortiums on the way now in the area of bioconvergence that would be active next year. We've done a lot around the area of human capital in this big world of bioconvergence to expose engineers to the area, to the biology concepts and terms. And we've built a bioconvergence community. We are building all kinds of courses around the bioconvergence for entrepreneurs and PIs and the intersection between them. We're very much encouraging academia and industry collaborations. We've opened a call with Korea, with South Korea, or with our mutual fund that we have with them, specifically dedicated for bioconvergence. Is still open up until the end of December for collaborations between Israeli companies and Korean companies. So we've done a lot of activities around this area, and as Itai mentioned, we have intentions to increase these activities over the coming years in the next decade, hopefully to build a cluster here, a leading cluster in this area. And of course, I think the most important building block in this cluster would be collaboration with countries around the world, and Denmark is really important for us. And so, as I mentioned before, I'm hopeful that this is only the beginning of a beautiful friendship. So thank you.
Thank you very much, Karina and Itai. We will now take a look at some applications of bioconvergence that are already happening in Israel. And so I would like to turn over the word to Yaniv Erle, who is the CEO of the Israeli startup 11TX. Please, Yaniv.
Thank you very much. Let me just share my screen with you guys. Okay, see my slides? Perfect. Okay, good. So good morning, everyone. And I would like to thank the organizers for inviting me to this very exciting conference. I'm the CEO of 11 Therapeutics. We are a therapeutic company, and our main aim is to combine synthetic biology with AI in order to develop better nucleic acid therapies. We started not a long time ago, but we are still in stealth mode. And so I kind of thought, what should I present you? And I thought to present you a research that I've done at Columbia University. I was a professor there. And this research inspired some of the aspects that we used in 11 Therapeutics. And I also think that the research I'm going to tell you about, which is about the DNA of things, is really kind of like a great example of bioconvergence, how we can bring computer science, material science, and also biology together and to build something quite new. So the story I'm going to tell you today starts in 1950s. And what you see over here, this is a hard drive made by IBM in the 50s. And this hard drive had 5 megabytes of data. And in the 50s, you need 6 strong guys that went to the gym in order to take this hard drive and push it into a huge truck. Now, fast forward to the present, we have these devices today, basically, that have more than 10,000 fold more data, and the size is 10,000 fold smaller. So this is a great revolution in our ability to store data. And without this revolution, we will not be able to live in this digital world that is so data intensive. But one thing in this revolution, if you think about all the storage devices, is that they all have very substantial geometrical constraints. In fact, the geometrical constraints of the storage devices determine how we call them. Let me give you an example. So a compact disc is a disc, right? That is why we call it a compact disc. If it's not be a disc, you cannot read information. It must be a circular format in a certain geometry. A tape looks like a tape. This is why we call it a tape. A hard drive is quite hard. You cannot bend it. So in all these devices, the geometrical constraints are so important that we name the device after its geometry. Now, if you think about it, kind of like if we think about the storage devices of the future, do we really want to be stuck with devices that have such rigid geometrical constraints? And if you take inspiration from nature and you think about DNA is kind of like, let's call it in the macromolecular level, is really in a liquid state. It can take any form, it can be in any order and so on. And DNA is maybe the ultimate storage device that we can think of. So let's think about that. Why DNA is the ultimate storage device. Whether you are a virus, a cucumber, an elephant, or Donald Trump, you store the most important information, namely how to make yourself, in DNA. And look at all these different organisms. They have various different shapes, and they all store quite a lot of information. So in human, we talk about 3 billion letters in each cell. And we can build various structures of the storage. We are not constrained in the geometry of the storage technology. In fact, if you think about it, it's a crazy idea to store information in DNA. I just want to remind you that if you look at the biosphere right now, the biosphere, the Mother Nature, stores 10 to the power of 37 bytes of information on DNA. So if you think about the milk in this coffee, the coffee beans inside, I have some dates here, they're all storage devices around me. 10 to the power of 37 bytes. Whereas all the human data, digital data that we store on hard drives, etc., is only about 10 to the power of 25. So basically, Mother Nature has about 12 orders of magnitude more data stored on DNA than we humanity made, and we call it big data, which is a bit ironic. Okay, so the question is how we store data on DNA. And the idea is that every file is basically just a string of bits, zeros and ones. DNA is also a digital molecule. It is composed of A, C, G, and T. So what we can do in a very naive approach, we do something more sophisticated, but just to give you an illustration, we could assign every 2 bits of information into a letter of DNA. And now if I can take these nucleotides and put them one after the other, I can represent my file as basically a sequence of nucleotides. How do we do that in practice? If we want to encode a certain DNA molecule, there are companies, they use inkjet printers. Instead of colors, they put in their nozzles DNA nucleotides. They fire these nucleotides on the same spot and it grows DNA chains. You can take these chains, it comes in this, actually this is a real example from my own research. You have many DNA molecules over here with all the data that you want. And once you want to retrieve the data, you just sequence it with one of your favorite DNA sequencers. So this is pretty great, and we published a paper in Science a few years ago. But now let's think about how we can use this technology to create objects, ordinary objects in any type that they can store information. And we call this idea the DNA of things, or DOT in short. And this was done in collaboration between my lab and the lab of Robert Grass and ETH Zurich. So here is the idea. We take a file, which is just zeros and ones, and we convert this file into DNA letters on the computer. And we write these DNA letters in the technology I showed you before using these inkjet printers. Now we take the DNA, we put it inside silica beads, we encapsulate it inside these like tiny glass beads. They preserve the DNA against environmental stressors. Then we take these glass beads, put them inside a polymer, some plastic polymer, and we extrude this polymer and create a long filament out of it. So now we have this like basically a plastic polymer with some DNA inside. And now we can take this polymer and we can use 3D printing to print whatever we want, any shape. So what you see over here, this is a student at the grass lab, and you see the lens of one of his glasses over here. This lens, which is from a plexiglass, which is just like a transparent material, we coated it with DNA, with silica beads with DNA, and we encoded an entire YouTube movie inside it. Now, if you look at the properties of this lens with the DNA inside versus this lens that doesn't have any DNA, they have identical optical properties. You cannot see that there is any DNA inside. So you can effectively conceal a video inside this lens, or a secret file if you want. Now, how do you retrieve the data? You take the lens, you just scrape a little bit of plastic material. And since DNA storage is so dense, there are so many bits per square millimeter, by just scraping a few tiny crumbles from this lens, you can extract the DNA, sequence it, and you can retrieve the file perfectly. And we encoded a few megabytes of information in one of these lenses. So this is a great way if you want to conceal some information and carry it with you. Let's say your Bitcoin passwords or some really sensitive data that you don't want anyone to find, you can just use it and create this lens. Now, we're not restricted to lenses or to transparent material. The buttons of my shirt, these plastic buttons, I can also encode file. I can encode a book in this button, I can encode another book in this button, and just carry it with me, and nobody. Oops, can you hear me? Okay. Okay, now I want to tell you about this bunny over here. Now, this bunny is a very special bunny. This is a 3D printed bunny. But it's not a regular bunny, because in this bunny, there are silica beads, and inside these silica beads, there is DNA. And this DNA encodes digital information about the STL file, how to print the 3D printed bunny. So what we have now is an object that carries its own blueprint on it. Now, how do you retrieve this information? You see this bunny, we chopped a tiny bit from the ear of the bunny, of this plastic bunny. We took this tiny amount of plastic, we dissolve it, and we retrieved all the DNA data from it. And then once we had the DNA data, we could produce new bunnies from it because we have all the information. We put in these new bunnies, the DNA from the previous bunny, and we created subsequent generations using the same process. And our encoding strategy is so robust that it can really tolerate errors in the synthesis and the sequencing of the DNA. So, and in fact, the last bunny over here, we waited 9 months, we retrieved the information perfectly and created a new bunny out of it. And we could continue the process, we just got bored by doing this process. And we figured out that we could create 10 to the power of 20 bunnies without any loss of information if we keep like chopping different parts of the bunnies and replicating them based on the amount of DNA that we have there. And that means that we can create these such bunnies like for the entire humanity for quite a long time. So the point in this process is now we can create objects that have their own blueprint, which opens quite interesting possibilities. So maybe if I can just summarize that with a short movie, I want to show you how we put all these things together with creating the bunny. So let's watch this movie together. So this is the scheme that we developed. This is the file of the bunny. We now encode it into DNA using a strategy we developed. We get the DNA oligos and we amplify them at the beginning. We now put the DNA into silica beads to protect them from environmental stressors. We put it into a filament, extrude the filament. And now we start printing the 3D printed bunny using the filament with the DNA inside. Now we chop a tiny bit from the bunny. That's enough to get all the information. We dissolve in order to get the DNA, and we just sequence it using a regular DNA sequencer that is found in various labs around the world, an Illumina sequencer. We get the data, we decode it, and we get back the file perfectly. Okay, so what this DNA of things is really good for? So we can create materials with blueprint, and that's quite important. We can create even electronic health records of individuals as a part of surgeries. When you put medical implants of these people, you can put the entire electronic health record of the patient inside this medical implant. So the patient will carry this implant with him all his life. So in the future, if you need to retrieve the information, you can just do a biopsy, get the synthetic DNA, sequence it, and know all this information. As I told you, it's quite great to hide information if you want to carry with you information to sensitive places. Not a problem. You can conceal any type, like this information, in virtually any type of plastic material. And we didn't develop a self-replicating robot, of course, but we did found a way to at least like one necessary but insufficient condition for a self-replicating robot, which is how to put a blueprint on an object. And this is a very good way to kind of like in the future, if we can also build DNA sequencers as part of these like materials, that would facilitate research in self-replicating robots. So with that, I'm happy to take any question. This research, as I said, was done with ETH Zurich and my lab at Columbia University. And we published a manuscript in Nature Biotechnology as me and Robert as senior co-authors about a year ago. Thank you very much.
Thank you very much, Yaniv, for this very inspiring talk of what the future will look like for us. And now I would like to pass the word on to Didi Dabush from MyBiotics in Israel, who will talk about what they are doing at the moment. So please.
Thank you very much. Hard to compete with what we just heard. It was amazing, very inspiring. I'll try to convince you that we need more than our DNA to be healthy. So as mentioned, I'm the CEO of MyBiotics, and I will try to show you how we can take bioconvergence into a very traditional industry and the way we are trying to change it. So MyBiotics is trying to close the gap between the discovery of beneficial bacteria and the clinically effective products. We all probably know about the microbiome, the effect of beneficial bacteria, but we understand that there is a need to change it. There is a huge gap between the potential and the actual ability to solve problems. So just a little bit about the company. The company is located in the Science Park in Rehovot. We have developed a list of technologies that provide us tools in order to develop good and more efficient product. We have a probiotic pathway, but in parallel, we also develop a clinical pathway towards drugs, which will be in clinical phase very soon. And just recently, we were awarded for Horizon 2020 in terms of next generation probiotics based on our bioconvergence approach and the ability to use new bacteria. I'll just give you 2 words about our clinical pathway or product, and then we'll talk a little bit more about the practice. So we're focusing on infectious diseases. There is a disease called C. difficile, where a complete microbiome, the whole bacteria of a human is changed using a human sample. And we are currently the first company that have been able to produce a complete microbiome composition. And this is going to clinical. We have an oncology program that is ongoing. We're very much focused on women's health and the ability to improve many aspects in this area, and also have the advanced probiotics approach, which we hope brings new changes and new capabilities to probiotics to actually have a clinical effect. I'll change it to this. I hope you see better. So for us, If you lived on Mars in the past 10 years, you probably didn't hear about the microbiome, but the microbiome refers to the bacteria that we have in our body. And it is an amazing number. We're talking about 10 times more bacteria than our own cells. And we're talking about 300 times more genes that can have an influence on our health. And the recent works shows that it has an effect on metabolics, on autism, on Parkinson, and many others. And with that said, still there is a huge gap between the ability to get this information and make it a product. Now I'm going to destroy your lunch and talk about feces for a second and stools, sorry. The only way today to treat or change the microbiome for patients is by taking a human sample, a complete human sample. And what you can see here, done by the way by a Danish doctor who is part of the company, is actually taking frozen human samples and giving it to patients with C. difficile, a very nasty disease that kills up to 70,000 people a year in the US alone. And try to imagine the FDA approving such treatment for patients. Why is it happening? There is, as I said, a huge potential, but a very big gap between the potential and the ability to create products. If we talk about the human microbiome, only 20 to 30% of the bacteria can be cultured. At the end of the process, passing through production and the human body, we remain with very, very small amount or diversity of bacteria, losing most of the potential to treat microbiome related indications. What is our magic? I have to say it was very simple at the beginning. We tried to think how, as babies, we are not sterile. We do get the bacteria to go through our body. They do survive everything. So we have been developing a method to grow bacteria on particles using their own survival and protection methods. Part of it is biofilm, it's a community, and it's a robust process, and it allows us to use very regular bacteria and give them the protection from extreme pH, such as acidity in the stomach, compression, heat when you want to use new product. But the most important thing is the ability to have real colonization and survival in the human body. Now I'm going to the interesting part of why bioconvergence is necessary in this field. This is a very traditional field, and Denmark, as you all know, leading this area. We started with the microbe technology, which is a single strain fermentation process. On the other hand, when I can show you the super donor. The super donor is a technology that allows us to take a human sample, a complete human sample, and using fermentation technology, get almost the same composition. So instead of a very small portion of a human microbiome, we can generate 500 treatments based on a good microbiome of a patient. But there is a huge gap between them. Only 30% can be grown when they are single strains. We can grow all of them when they are growing together. We needed to understand what's happening in between. So this is where we started the Super Donor Select, which is the cluster-based composition of bacteria. We wanted to understand why specific bacteria requires other bacteria to generate or give it the environment and condition it needs. And in order to do that, talking about thousands of bacteria, we needed to understand better what are the relationships. And this is the MyLiving technology that involves AI, culturing processes, interactions between bacteria, and a system that can help us understand the interactions between the bacteria in real life in the lab. I'll give you two examples of that, about the ability of the technology to go and become a product. I mentioned the tolerance to pH, but we can also, using specific materials and formulation that were also developed in the lab, make these bacteria survive heat and then create new products and new abilities for existing probiotics, but also make more stable products, such as you can see here below. We integrated bacteria that are very known into dairy, into non-dairy product, and showed that they are much more stable. And this is a combination of the chemistry, the biological, and the understanding of the bacteria. Few words about the super donor technology. So the super donor technology is a system that will replace the whole microbiome treatment using stool samples. We have been able to culture and grow the whole population in an industrial process and get to up to 90% similarity to a human sample. We took this opportunity and we have now the ability to produce artificial human microbiome and then be able to use them for different purposes. Let me jump and show you what's been done today. Today, because of all the gaps that I mentioned, companies that are already in phase 3 in clinical trials still use human samples. They generate better safety tests and better screening capabilities, but no one can actually grow all of them. We've been able to show that we can grow them using our deeper understanding of the interactions between these strains. This is my favorite. The SuperDonor Select is really taking us to the next step. When we talk about the thousands of strains, and we would like to take very significant bacteria that have an effect, for example, on biological treatment for oncology, and we want to grow this bacteria and make it a product, we need to understand what is the environment that needs to surround it. So there is a nice example about the bacteria called Prausnitzii, very hard to grow. We've been able to show using our computational capabilities, what are the interactions, what are the cross-feeding mechanism that this bacteria has, and created a group of 8 additional strains that support this bacteria, not only in the culturing process, but also during colonization in the human body. Another layer which is unique, and at the end we'll combine everything to one approach. Another thing that is very unique is the ability to predict how the human microbiome will behave when introduced to a new material. We created a small high throughput system that keeps the whole microbiome viable. We can put drugs or materials inside this environment and check the differences or the effect on the microbiome of the patient. We've just recently showed it with a metabolic disease, a drug for metabolic disease. But we're taking this to the next step, printing 3D printing of a gut that represent how these bacteria behave in the human gut, trying to understand the effect of different materials on the human microbiome as a whole. So trying to wrap up everything, I have 2 minutes. I described the artificial intelligence, understanding the behavior of the bacteria, the screening system, the artificial donor design bank, all together takes us to an approach that I think is very unique in this microbiome area. We would like to get into our system, actually this is what we're doing, any type of microbiome data from patients, from research, from in-house discovery processes, run through a development process and combine it with our delivery, while we can create fast cycles between the discovery towards a product. We also introduce our technology to researchers, allowing them to have a complete microbiome recovery or change using our system towards a product. And now for our Danish part. 4 years ago, I met Christian Brøndby, who is here as well. He was very senior in the probiotics and the ecosystem in Denmark. And it was a huge jump for me, understanding how in Denmark this field is so much innovative, strong, but in the same time go from idea to a product. And this is something we are trying to learn as well. And we're doing a lot of efforts in the past 3 years, approaching companies in Denmark, including trying to bring investment into Israel. I think there is a huge connection between the two countries. And also our medical advisor was born in Denmark, Ellen Jungster, and I think this is a great lesson for us as a company, how to go from an idea to a product. And thank you very much for the opportunity today.
Thank you very much, Didi. That's very inspiring, and it's great to see there's already a collaboration going on that we can build upon. Now we will open up the panel discussion. So if anybody has any questions, you're welcome to post them in the chat. However, first I would like to just introduce Bjarke Christensen from the Technical University of Denmark, and we've asked him to just say a few words about what they're doing at the Technical University, one of our leading universities in this field in Denmark. So please, Bjarke.
Okay, thank you. Thank you very much. I think thank you for inviting me, first of all, and also thank you for some extremely inspiring talks, both about the Israeli strategy on bioconvergence and also some really inspiring examples. So thank you for that. I have to admit that bioconvergence is not a word that I was very familiar with before I talked to Louise. But maybe we should, not only within the health area, but actually within the sustainability area. I see huge potential. But actually, I also think we do quite a lot. I think maybe all the university regions here in Denmark have really strong life science environments, including university hospitals and relative strong engineering environments, where we actually have the possibility to combine the biology, digitalization, and more hard technologies, both in research and innovation programs, but also in research and innovation centers or units. And then, of course, as Søren also mentioned, adding the strong life science industry here in Denmark, I can only say that we have a potential here really to create a bioconvergence research and innovation environment. I also think that we have funding bodies that support this multidisciplinary activities going, and maybe going even broader than the technology, and also including things like social sciences. Also our mission-driven programs, as we are in the process of establishing in our national center, or in our national attempts to, for example, deal with the CO2 emission and green challenges, will undoubtedly aim for multidisciplinary programs. Also, we have some private foundations that have relatively large programs supporting projects where data science combines with specific disciplines in health tech and sustainability. So coming back to Louise's question, what are we doing here at the Technical University? In fact, I realize we have actually done a lot in the last 5-10 years in the life science area. First of all, life science constitute nearly 30% of the research at DTU, research and innovation. Also, we have done quite a lot to bring disciplines together and in fact work exactly with what you call bioconvergence. 2 years ago, we established the DTU Health Tech department, and this department brings together scientists from electroengineering, nanotechnology, bioinformatics, immunology expertise, vaccination expertise, animal facilities, and diagnostics into one big department. And of course, that has a huge potential for making bioconvergence research and innovation. And I see that it will probably all be about bioconvergence. And that actually also goes for my own department, D2 Bioengineering, where we basically try to use the molecules of light to engineer solutions for health and sustainability. In recent years, we have moved from being mainly a biology-driven department to combine and develop both hardware and software technology with more life science-focused research, such as cell factory engineering, fermentation, enzyme engineering, microbiology, and many other things. So again, we go mainly from biology focus to let biology and technology go hand in hand for new research and innovation development. So I think that actually tells a little bit about that we are really doing a lot in this multidisciplinary life science innovation area. But I think we can also do even more if we get a more national strategic focus. That will probably even enhance it more. And in fact, very recent we have established a new life science cluster here in Denmark with the aim to actually connect the biotech, the pharma, and the medical and the welfare tech area in one big national cluster. So not just going across here in Zealand and in the southern Sweden area, but actually going across the entire Denmark. And one of the things we really want is to create a bridge between research institutions and industry. And actually with this cluster, I think we can push this bioconvergence agenda even further. So yeah, that was my words.
Wonderful. Thank you very much, Bjarke. It's very encouraging that there's so much going on in both countries and also still a lot of potential to do more. So time flies when you're having fun. So I think we will just ask one question to each of the speakers today, which really is, if you can each just name one actionable takeaway, something that we can do today to drive multidisciplinary innovation, as we all like to see action happening. So maybe I will start with Ambassador Rigelsen. If you have one takeaway from today that you would like us to consider.
Thank you very much, Louise. I must say, time really flew, and not every Zoom meeting is as fascinating as this one. So first of all, I'm very grateful that it was so inspiring and really educational. My takeaway is that there's so much in this world where we, as relatively small countries with a healthy dose of self-esteem, can tackle. And I loved the way that we both looked at what is necessary at the research level, but also jumped up to what is necessary when we look at what state and sometimes very sad state of the world we are finding ourselves in. COVID is one warning signal. We heard from Bjarke and others that we have a climate crisis that we would like to ignore because it is so great that it will let us lie awake at night. But we have to tackle it, and we have to tackle it in our lifetimes. And then, of course, we have in many ways a human disconnect between our countries, which can also make this world drive into the ditch and not on the highway. And I think combining what is done and the great progress and the great sense of optimism with the great challenges is what we need, and is a takeaway from me. A takeaway that the next time I sit in a panel with foreign policy wonks and nerds, I will invite one of you, because if not, I think we will not really hit the road in the way we should. Thank you.
Thank you very much. Karina from Israel Innovation Authority, an actionable takeaway from today?
Yes, just to resonate to what the ambassador just said, we are really aiming at building a strong industry leadership here in Israel. I think this is a very good ground for collaboration between the two countries to tackle the next wave of whatever crisis there will be. Hopefully not, but pandemics and other crises we are facing, and there are huge challenges that need to be faced. And the current the current way that we are working is probably not as precise as it should be. And again, just to elaborate a bit more about what we are targeting at Bioconvergence, at least, we're looking at, again, the intersection between biology and engineering to provoke innovative solutions. And that can be, just to connect it to the climate crisis and that. So that can be also in the areas of energy, in the areas of agriculture, in the areas of food, in the areas and other areas that are not just merely health tech solutions. I think that it's really a broad world of solutions that can be, at the end of the day, tackled with the help of the collaboration between the two countries. And I think it's a very good ground to start with.
Thank you very much, Karina. And Yaniv, from an industry startup point of view, any actionable takeaway from today?
Yeah, I think that to get bioconvergence, one of the major things to do is to break the silos we have in academia. We have silos like people are, we train them, oh, you're other computer scientist, or you're a biologist, or you are an electrical engineer. If we really want to unleash the power of all these technologies, I think we need to give people the ability to be trained in multiple fields and to embrace that. There is, I think, in academia, a lot of hostility against multidisciplinary research. People like their own fields. They don't accept newcomers, and this needs to be changed in order to get really to put all these things together.
Thank you very much, Yaniv. And Didi, your takeaway?
I can't agree more with what Yaniv just said. I think what is the motivation today is not being a biologist or doing a specific research. It's a need. We need to target a need and take all the tools that we can find in the academia, in different places. I learned a lot about the ecosystem now in Denmark, and I think it's a very strong environment that can bring beautiful solutions to the needs that we're facing.
Yes, thank you very much. And Bjarke, your takeaway?
Yeah, I think, I have to admit that sometimes you shouldn't invent the wheel again. And I have to say, I really take a lot of inspiration from this meeting in terms of the Israeli strategy on bioconvergence. This is something we should think about in Denmark. And I'm totally agreeing with Yaniv on this silo thing. We have to develop cultures around this that creates an environment where biology goes hand in hand with engineering. That's really a key thing. And I think one of the things we could think about is this, that, we have journals that are extremely hypothesis driven. Make them hypothesis driven in combination with technology driven. That would really change something, because that's the impact thing here. So, I think there's a lot to take from this meeting. Thanks a lot.
Thank you very much. I think we have some very tangible
if I may add some take on from
Yeah.
my side. So I would like to emphasize that the challenge, the real challenge, is to build sustainable industry. So this was what we start in Israel. The thinking we were highly frustrated. We have a great life science research and great healthcare system in Israel, but we didn't succeed to build a sustainable life science industry in Israel. It could be compared to the ICT industry that we succeed to build in Israel. So the idea is how to leverage, the rationale was how to leverage the research and the technology to build industry. So we need to leverage and to develop new talents in the academia and in the industry to understand both engineering and biology. And these talents will be the basis for to build these new ecosystems. It's very important to emphasize that we all have great research, academic research, but we need to the need and the vision is to build a sustainable bioconvergence industry with high economic impact. This is the main aim. Thank you very much.
Very good points from all of you today, and some very tangible actions for us to take with us and see what role we can play in advancing these in order to really benefit from a field that can change the lives of so many patients and populations to improve our healthcare in the future. And so with this, I would like to just give the word to my colleague Eldim at our consulate in Istanbul to round off this webinar today.
Thank you, Luza. I hope you can hear me. On this occasion, I would like to thank all our distinguished speakers and valuable participants. Our region has a lot of opportunities and public drive for establishing partnerships to implement a successful and functioning digital healthcare technologies. I would like to invite any interested Danish stakeholders out there to get in touch with the Trade Council in Istanbul and the Innovation Center of Denmark in Tel Aviv to explore the possibilities of working together. You can find out our contact details on our websites, Denmark in Turkey and Denmark in Israel. This was the last strategic webinar series on digital healthcare facilitated in partnership with Innovation Center Tel Aviv and Trade Council in Istanbul. And thank you for participation, and we wish you a wonderful Christmas break. Thank you.
So thank you, Louise, again, and a lovely Hanukkah also for everybody.
Yes, happy holidays to everyone, and thank you for joining.
Bye, have a lovely day.
Bye.